Now, in the biological molecule chapter, which is chapter 2, we have looked at carbohydrate and lipids. Today, we're going to start to look at the protein structure and function. So the learning outcomes of protein includes describing the amino acid structure, formation and breakage of peptide bond, describing the primary, secondary, tertiary, and quaternary structure of proteins, describing the interactions that hold protein molecules into the shape. And the last one is we will describe about globular proteins such as hemoglobin and fibrous proteins such as collagen. Now, the monomer of protein is amino acid. Let us look at the structure of amino acid. Now, amino acid has a central carbon, okay, which is also called an alpha carbon. So usually when we name or number the carbon in an organic molecule, carbon nearest to the functional group is always carbon number 1, followed by 2, 3, 4, 5. Sometimes carbon number 1 is also labeled as alpha carbon. So in this case, we also call it as an alpha carbon. Now, carbon has got 4 covalent bonds. So therefore, there'll be 4 atoms or 4 groups that can be held by this carbon. So let us look at what is being held by this carbon, the central carbon. So to the left of the central carbon, we have got the NH2 group, which is the amine group. Now, in amino acid, we can also call this an amino group, okay, because amine group is a functional group of the amino acid. So we can call this NH2 amino group as well. It is basic. So to the left side is NH2. Now to the right side is actually COOH group, which is called the carboxyl group. Now any molecule which contains the carboxyl group can be called the carboxylic acid. So in amino acid, this is also called carboxylic acid group. So it is located to the right side of the central carbon. Now on top of the central carbon is actually the R group. R group stands for the variable group. Now, variable means can be different, right? So why is it called a variable group? Because we know that there are 20 or more amino acids in our body. So R group, different amino acid will have got different R group. So this R group or variable group will determine the identity of the amino acid. That means it will determine what amino acid is it. So it will be located on top of the central carbon. So the last element is at the bottom is the H. So this is the overall structure of the amino acid. So very easy to remember. Amino group, acid group, okay, or amine group, carboxylic acid, or carboxyl group. So amino acid. Okay, very easy to remember. So the amino acid molecular structure can also be drawn in red color form, where it shows you where the H and the O and OH are being located in the amine group as well as the carboxyl group. And this amine group and carboxyl group can be charged as well, which I'll show you in the next slide. Now, by the way, a molecule which has both the basic and acidic group is also called amphoteric. So amino acid got the amine group, which is basic, and the carboxyl group, which is acidic. So amino acid is considered as an amphoteric molecule. Now let us look at this structure here. Now this is an amino acid. When we add acid to the amino acid, the acid will react with the base, which is the amine group. So when the amine group react with the acid, it will take up the acid, right? The base will take up the acid and becomes NH3 plus. So the plus sign comes from this acid because it has taken up this acid. So from H2 becomes H3. And the positive charge comes from this positive charge. Okay, so NH3. Now, what if we add a base to the amino acid? When you add the base to amino acid, the base, I represent it with OH negative here. So the base will react with the acid part of the amino acid, which is the H, and takes this H away, right? So when this H is taken away, this H is a H plus. H plus and is taken away. So when this H plus combined with OH minus, it becomes H2O. The plus and minus sign will cancel off each other, so it becomes neutral. So when you take a plus away, here becomes negative. Right? So it becomes COO negative here. Because the base has taken the acid away, it has reacted with the acid. So what's left here is COO negative. So what you can see here is an amino acid can react with both acid as well as base. And then when it reacts, it will be a molecule having both the positive charge as well as the negative charge. So this kind of molecule is called a Zwitter ion. So Zwitter ion is a molecule which may have both positive and negative charge, but overall electrically is neutral, right? Because it carries one positive charge and one negative charge, so it will cancel off each other. Overall, the molecule is neutral. Okay, this molecule is called dipolar. Polar means having pole. So you got a positive pole here, and you got a negative pole here. And this pole has got the same magnitude. So you've got one positive charge on the left and one positive charge on the right. So that's why it's called dipolar. That means having two poles of two different charges of equal magnitude. Two poles, two different charges, equal magnitude. So because its charge is soluble in water. So this is to show you that amino acids can be charged, right? Now, amino acid can be a buffer and an ion. Now, what is a buffer? Buffer is a solution that can resist the change of pH upon the addition of small amounts of acid or base upon dilution. So what I've shown you just now in the previous slide here is that whenever you add a small amount of acid, now this acid will then react with the base to become NH3+. So therefore, it will not change the pH of the solution because the pH of the solution is actually proportional to the concentration of H+ in the solution. So once you add the H+ into the solution, the H+ already reacted with the NH2, it becomes NH3+. So this acid will not reduce the pH of the solution anymore. Now, in the same way, when you add the base into the solution, now the base will straight away react with the acid to become water, and water is neutral. So again, this base will not be able to increase the pH of the amino acid solution. So because of that, whenever you add a small concentration of acid or base to the amino acid solution will not change in the pH. So therefore, amino acid is considered as a buffer solution. So buffer solution again is a solution that resists the change of pH upon the addition of small amounts of acid or base or upon dilution. Now let us look at the R group. R group is called the variable group. It determines the identity of an amino acid. Now, there are four major groups of R groups. First one is neutral, non-polar, so it's hydrophobic. Second one, neutral means the charge is neutral, but it is polar. It means there's uneven distribution of charge. So it's hydrophilic. Third one is basic, which can be charged, so it's hydrophilic. Fourth one is acidic, which can also be charged. It is hydrophilic as well. So the first group is neutral, non-polar. Non-polar means there are no uneven distribution of charge. Overall, it's a hydrocarbon skeleton arranged in chain form or in ring form. So you can see that over here, this is the this overall amino acid. So you can see this is the central carbon, right? This is the amino group, and this is the carboxylic acid group. So amino acid. Now this is the R group here, and this is the H, right? Now this R group is a H. So amino acid with the R group H is called glycine. GLY, the three-letter abbreviation, this is the abbreviation of glycine. Abbreviation means the short form, okay? Now, the rest of them, you can see, now this is also a hydrocarbon. This is also hydrocarbon. This is a hydrocarbon with SH. Okay, so this one you need to remember. Okay, these two you need to remember. Glycine is an amino acid with the R group is a H. So this is the smallest R group, which is made of H. Cysteine has got an R group made up of SH. Okay, so this one you need to remember. The rest no need to remember. Just know that they are all hydrocarbon here, right? These are all hydrocarbon, or the benzene ring, right? This is called the benzene ring. It's a hydrocarbon ring, okay, with double bonds in it. So this is non-polar R group. Next, this is the polar R group. It's still neutral. So remember, polar means uneven distribution of charge. So you can see that this group here, OH, is polar, right? But it's overall no positive negative charge. So all these are polar, right? This is polar. This is polar, okay? This is also polar, but overall no charge. Okay, overall no charge. Even though this is delta negative, remember you learned already this is delta negative, and H plus, this is delta positive, but overall there's no charge. So it's polar, uneven distribution of charge in the R group. All right. Now you don't have to memorize the name of the R group. You don't have to memorize the structure. You just have to know there are 4 types. Okay, and you know how to identify which type they are. Now over here, there's basic R group. So basic R group has got the R group has got the functional group, which is the amine group. Okay, so it can be charged because it's basic. When you take up the acid, it will be charged. Right? So therefore, when it's charged, it is positively charged. Now on the right side, this is an acidic R group because it's got the carboxyl or carboxylic acid group. So when the H plus reacted with the base, whatever left is COO negative. So therefore it can be negatively charged. Okay, so learning check. So over here, select from the above structure which one is the alanine in base. Alanine in base, that means it will react with the acid, right? Okay, so that means the acid H plus from the COOH will react with the base, and when it has reacted, the COH becomes COO negative. So therefore, the answer is 2. Now, alanine in acid, so when there's an acid, acid will react with the base. So acid will react with the base of the amino acid. So the basic part is the amine group. So when the acid react with the amine group, it will combine with the amine group and becomes NH3+. Okay, so alanine in acid will look like the structure in number 1. Okay. Okay, now we're going to look at how we join the individual amino acids to form a dipeptide and continue to add amino acid to form a polypeptide chain. So over here, what you can see is you have got one amino acid here, okay, and you've got another amino acid on the right side. Take note that because it's different amino acid, therefore you need to label the R group as R1, R2, and so on. Okay, so name them. This is amino acid number 1 and amino acid number 2. And the reaction is condensation reaction. When you combine them, it's condensation reaction. And take note where the water molecule is being removed. It is being removed here. Okay, OH and H. So this is the only location where the water can be removed. So OH from the carboxyl group of amino acid number 1 and H from the amine group of the amino acid number 2. So this will form the so-called water molecule. So remember to write the water molecule is removed from condensation reaction. And then the C here will be able to form covalent bond with the N here. So that's where the covalent bond is being formed to connect the 2 amino acids together. So the new bond form is called the peptide bond. So you need to circle both the C and the N, or you can also circle this whole thing here. So this whole thing here is called the peptide bond. Okay, so this molecule now is called the dipeptide. So having 2 amino acid, dipeptide. So when you want to break them away to form back the 2 amino acid, you must carry out hydrolysis. So lysis means to break. Hydro means after you break them, you use the water, hydro means water, to seal the wound over here and here. Okay. Now, if you want to continue to extend the chain and make it longer, you need to continue to add amino acid to the right side. Okay, add to the right side one by one. So every time you add, you have to remove 1 water molecule by condensation. So therefore, condensation reaction to continue to add the amino acid. So if you add n number of amino acid, you have to remove n number of water molecules. Now, in the same way, you can break down the amino acid by hydrolysis. Sorry, you can break down the polypeptide chain by hydrolysis to remove the amino acid one by one. Okay, so remove them one by one. You can do that. So this by hydrolysis reaction, and then use the water to seal the wound. Okay. Now over here, I actually show the star. So how to draw the polypeptide chain? First, you must know what is in the backbone. The backbone is made up of NCC. Remember, amino acid is N on the left side, NH2, and a central carbon. So the second one is C, and the third one is the carboxyl group. So it's NCC, NCC. So the backbone of amino acid is always NCC. So here, can you see here? N, okay, C, and C. So this is one amino acid residue. And this is N, C, C. So this is the second amino acid residue, followed by the third, and so on. So this is one amino acid residue, NCC. And this is another amino acid residue, NCC. So can you see the star that I have written there? So the star denotes the covalent bond that join between the 2 neighboring amino acid by condensation reaction. Okay, and this is the peptide bond. This is the peptide bond. Okay. Now the next thing is, remember to label the polypeptide. Now when you look at the entire polypeptide chain, at one end of the polypeptide chain is NH2, and the other end is always COOH. Therefore, we can say that in a polypeptide chain, there are 2 ends. One of the end is called the N-terminus, and the other end is called the C-terminus, right? The C-terminus. So same here in this polypeptide chain, you've got N-terminus and C-terminus, or you can also name them as N-terminal and C-terminal. Okay. So this is a homework. please draw this 3 amino acid that has joined up to form a tripeptide. And the 3-letter abbreviation for this will be, for this tripeptide is alanine, glycine, and serine. Now, you don't have to memorize the short form, okay? You don't have to memorize. You also don't have to memorize the R group. You only need to know there are 4 types of R group, and when given the structure, you know how to identify them, which of the 4 R group is it. That's all, okay? Now, let us go and look at the conformation of polypeptide. Conformation of polypeptide. Now, conformation, this word means the 3-dimensional structure or the overall shape of the protein. Conformation of protein. Okay, so there are 4 levels of protein structure that we can look at. We have got primary structure, secondary structure, tertiary structure, and quaternary structure. Now, primary structure is very easy. Primary structure is the sequence of amino acid in a polypeptide chain. For example, now in this short polypeptide chain, over here you can see the sequence of amino acid where the first one is alanine, followed by thyroxine, histidine, leucine, valine, and glutamine. Right? So this is the primary structure of amino acid. Now let us look at the second example. Now this is an insulin molecule showing you the primary structure of insulin. So you can see the amino acid sequence shown here very clearly. Now it always starts from N-terminus as amino acid number 1. So this is N-terminus of this polypeptide chain. So this is amino acid number 1, right? This is C-terminal of this amino acid chain, polypeptide chain. So this is the last amino acid. Now, insulin is made up of 2 polypeptide chains. So the second polypeptide chain is called chain B with 30 amino acids. The first chain is chain A. It's named chain A. It's got 21 amino acids. Okay, you don't have to memorize, just look at it. Now, N-terminus. So this will be the first amino acid. And in C-terminal, this will be the last amino acid. So this chain shows you the sequence of the amino acid. So this is the primary structure. Sorry, not sequence of amino acid, sequence of the amino acid in the polypeptide chain. So this is the primary structure of insulin. Now this is a lysozyme also showing you the primary structure, means amino acid sequence of the lysozyme. So again, here is the N-terminal or N-terminus. So this lysine will be the first amino acid. Now, this is the C-terminus or C-terminal. So this leucine will be the last amino acid, altogether 129 amino acids. So now we're going to look at the secondary structure of a protein. So secondary structure is the spatial arrangement of a polypeptide chain. That means you've got this polypeptide chain. In this space here, how does this chain arrange to give you an overall 3-dimensional structure? So in secondary structure is a spatial arrangement of this polypeptide chain due to regular coiling, means like this. So this chain coil up regularly. That means every coil is the same, so it's called regular. Okay. Or regular folding, means this chain starts to fold this way. And it folds regularly, the pattern is the same in every folding. Okay. So these 2 structures are called the secondary structure. Now, the coil or spring-like structure is named as alpha helix. And the folded structure, regular folding structure, is called the beta-pleated sheet. So this is the beta, okay, dash pleated sheet. Okay. Pleated sheet. Pleated means folded. Okay. So these are the 2 forms of secondary structure. The two types, alpha helix and beta pleated sheet. So then let's look at the detail, how this structure is formed. First, alpha helix. So what you can see on the right-hand side is actually the polypeptide chain, and it's showing you very clearly the individual amino acid residues in the chain. Can you identify them? So can you see this is the first amino acid because you've got the NH here, amine group, and the central carbon, and then to the right side of the central carbon is the carboxyl group, so amino acid. So this is the first amino acid residue. This is the second one. This is the third one, and so forth. You've got the fourth one here. Hang on. Okay. Okay. Hang on. So you got the fourth one, the fifth one. Okay. All right. So how does this entire chain arrange to form the coil shape of alpha helix? Now, can you notice the red color box I've drawn here? What do you think is this red color box here? What does it represent? Remember, what does it represent? Now, this represents the peptide bond that join the two amino acid residues together. Can you see that? This is a peptide bond. Okay. Now, the carbonyl group, this one here, okay, and the NH group, both of them are actually polar because there's an uneven sharing of electrons between the C and the double bond O, between the N and the H. Now, remember when you study the hydrogen bond, I mentioned before that the hydrogen bond is the bond formed between electronegative O. So let me show here. Okay, it can be hydrogen bond formed between the electronegative O and electronegative H, OH, NH, FH, remember? FH. And of course, there's one more, which is actually C double bond O. Okay. So C double bond O. Okay. So I've shown you before that the OH, NH, FH, right? All of them got uneven sharing of the electrons, that the O is more electronegative because it takes a bigger share of the electron. So there are more electrons here. The H tends to be more electropositive because it has got less electrons here. So unequal sharing of electrons. So here also same, the O takes more electron share, and therefore the C takes less of the electron share. Okay. So therefore, hydrogen bond can be formed between a delta positive H with a delta negative O and F. Okay, and this O as well. So over here, we can see that this O is delta negative, right? And this H is delta positive. So therefore, there's a possibility that they can form hydrogen bond, isn't it? So what happened is like this. So imagine that every amino acid is a family, okay? Is a family. Or let's say, imagine each of these amino acid is a person, right? So they line up together. Now, amino acid number one, it wants to form bond with one of the amino acid. So you have to look around and see which amino acid can form bond. Now, every amino acid, it is actually able to form hydrogen bond because every amino acid got a delta positive H, isn't it? Here, okay? Delta positive H here, right? So therefore, actually, this delta negative O can form hydrogen bond with any of the delta positive H in every amino acid. Now, in alpha helix, the first amino acid, okay, the delta negative O, the first amino acid decided to form hydrogen bond with the H, delta positive H of the amino acid that is 4 amino acids away. 1, 2, 3, 4. So it have decided to form a hydrogen bond with the delta positive H, which is located 4 amino acids away. So which means to say amino acid number 1 will form hydrogen bond with amino acid number 5. So you see, when they want to form the hydrogen bond, it means they want to hold hand. So when they want to hold hand, they have to come near to each other, then only they can hold hand, right? So therefore, from a chain that is very straight like this, when this part here decided to form bond with some part further away, now the entire chain will have to bend already, isn't it? So it has to actually bend in some way to bring this part close to here, this point here, then they are able to form the hydrogen bond represented by 3 dashed line. Can you see that? So that's why the entire structure already changed already. So it starts to bend. But don't forget, now the second amino acid also decided to form bond with the 6th amino acid. The third amino acid decided to form bond with the 7th amino acid, right? So therefore, they have to keep on moving until the whole chain becomes coiled up like this. Why they coil up? Because of the movement of the chain to allow hydrogen bond to be formed between the amino acids. So that's why it becomes coil shape, right? This one have to move here. This one have to move here. So the whole thing becomes coil shape. And then you can see there's extensive hydrogen bonds in the entire chain to hold the chain into this spring shape. So this spring shape will not change. So this spring shape is called the alpha helix. It's a name given to this structure. Okay, it's fixed. It will not change shape because of the hydrogen bond. Okay, so let's recap. The electronegative O of the carbonyl group, okay, the CO is called the carbonyl group, forms hydrogen bond with electropositive H of the amino group. So this electropositive H of the amino group, right, located 4 amino acid residues ahead. So this is the 4th one, okay, 4 ahead in the linear sequence, means in the straight chain, right? You count 1, 2, 3, 4 in a linear sequence, the 4th one ahead, okay? So that is amino acid number 1 will form hydrogen bond with amino acid number 5, amino acid 2 with amino acid 6, amino acid 3 with amino acid 7, and so on. So you have to know that the hydrogen bond is formed between both the CO group and the NH group, which also are part of the peptide bond, right? So this NH is part of the peptide bond of this, and this CO is part of the peptide bond of this one here. Okay, so that's where the CO and NH are located, is inside the peptide bond and is able to form hydrogen bond as well. Okay. So this is the overall 3D structure after the hydrogen bonds are formed among the amino acid residues. Okay. So due to the extensive hydrogen bonds, alpha helix is very stable and rigid. Rigid means you cannot bend one. It's like very, very hard. You cannot bend it, okay, rigid. Like a rigid person means it cannot change, cannot bend, and it's rod-like, okay? Now, in order for the chain to coil up like this, it must make sure that the R group is small, okay? The R group in the alpha helix must be small. Why? You can see here. Now, if the R group becomes very big, like this, if the R group is that big, take up this space, can you see that this hydrogen bond can no longer be formed? Can you see this hydrogen bond here connecting between this O and a H? This one cannot be formed already. So whenever hydrogen bond cannot be formed regularly in every amino acid residues, then the polypeptide chain will not be able to fold into its alpha helix. So therefore, in an alpha helix, all the R group must be small enough so that it will not block the formation of hydrogen bonds between the CO and the NH within the backbone of this polypeptide chain. Okay. Now, 1 turn of this coil is made up of 3.6 amino acid residues. So that means when your eyes look from the top, okay, it makes exactly 1 complete turn. All right, that means you see 1 complete turn from the top here is actually 3.6 amino acid. Okay, so example of alpha helix in our body. Now, alpha helix is like coil, so it's fibrous. It's a fibrous structure. So it's found in the keratin in skin cells. So it's used to make keratin in skin cells. Can you see it's very fibrous? Can you see the hole in the center here? This is actually the nucleus of our skin cell. Then the cytoplasm contains a lot of the keratin, right? Provide structural support on our skin. Now, we also contain keratin in our hair. Can you see the fibrous rod-shaped keratin in our hair? Also provide structural support. So it forms a structure on our body. Now, these are other alpha helix in the form of keratin, which also provide structural support to other animals. So you can see nail and skin, feather, horn, claws, hair, hooves, beak. Can you see hooves and beaks are really, really hard? So it's called rigid. Wool, okay? So these are examples of keratin in animals, which is made up of alpha helix. Now let us look at the beta-pleated sheet. So what you can see here is the two polypeptide chains arranged parallel to each other. Right? Can you see here? There are 2 polypeptide chains arranged parallel to each other. And can you see the arrow here? Now, do you know what does this arrow represent? Can you see or not? Now, this arrow actually represents the direction of the polypeptide chain. So what do you mean by the direction? Remember, when you add the amino acids to form polypeptide, you always add from N-terminus to C-terminus. So the chain always grows from N-terminus to C-terminus direction. So can you see here? For this direction, that means to say that this is how the chain will grow in length. So this must be the N-terminus and this must be the C-terminus. See whether it's correct. So that means this must be the N-terminus, right? Which is correct because it's N, this is C, this is C. So NCC is arranged like this. So this is 1 amino acid residue. Can you see here? This is 1 amino acid residue. Can you see? And then this is the next amino acid residue, NCC. So can you see that N is on the right side and the C is on the left side? That's why the direction is from the right to the left. So in the same way, at the bottom chain, the NCC is arranged this way. Can you see not? NCC. So the N is on the left and the C is on the right. So the direction of this chain is the N is on the left here. And the C is on the right. So that's the direction of the chain. Okay. So in a beta-pleated sheet, we have got polypeptide chain arranged side by side, parallel to each other. Okay, parallel to each other. And the arrangement of this parallel chain can either be of the same direction or opposite direction. Okay, you know already what same and opposite direction, right? Same means this is N-terminus and this is C-terminus. And then this is also N-terminus and this is the C-terminus. So this is called same direction. All right. Okay, different direction means it's different, like this one here I've shown is different direction. Okay. So when they arrange parallel to each other, again, they'll be held together by hydrogen bond. So the hydrogen bond is the same, it's a bond the hydrogen bond is a bond formed between a delta positive H of the NH and the delta negative O of the C double bond O. Okay, delta positive of the NH and the delta negative O of the C double bond O. So therefore, this is where the hydrogen bond is being formed, right? Hydrogen bond is being formed, and all the CO and NH are part of the peptide bond, as you can see here. Can you see this is a peptide bond? Can you see that this is a peptide bond here, okay, in red color, okay, right, okay, so NCC, okay. Now this is also a peptide bond. The N is missing here. I was looking for the N. The N is here, okay, there's an N here. So again, the hydrogen bond is formed between the CO and the NH, which are also involved in forming the peptide bond. Okay, apart from forming peptide bond, they can also form the hydrogen bond. Can you see here? The hydrogen bond. The hydrogen bond. So hydrogen bond overlap with the peptide bond. Okay. So to describe in words, the polypeptide chains are held by hydrogen bond between the electronegative O of the carbonyl group, the CO group, of an amino acid residue with the electron positive H of the amino group, the NH group, of an amino acid residue in the adjacent chain. Adjacent means neighboring, okay, neighboring chain. So what you can see here is, can you see this is a single polypeptide chain which can bend and fold to form, which can actually fold, and the parallel chain can form hydrogen bond across one another to hold it into a structure called beta-pleated sheet, right? Beta-pleated sheet. And even the direction is shown very clearly. Direction of N to C terminus is shown very clearly. So this is the structure of a beta-pleated sheet shown in this diagram here. So this part here of the chain is called the beta-pleated sheet, right? So therefore, when you draw the chain of the beta-pleated sheet, it will look like this, okay? So like you take a piece of cloth and then you fold it this way. So beta-pleated sheet, okay? And this structure will remain always like this because the neighboring chains are already held together by extensive hydrogen bond. Can you see how many hydrogen bonds are here in this short length? Already got 1, 2, 3, 4. So if it's very long, it's got extensive hydrogen bonds. So the whole structure will not change one. It's fixed, regular folding into beta-pleated sheet structure. Okay, so this is the beta-pleated sheet structure in 3D structure. So like a piece of cloth that is folded or double folded onto itself. So it folds onto itself. Okay, this is called single fold, right? Correct? Not this single fold. This is called double fold, right? So keep on fold onto itself. Okay. Okay, so due to the extensive hydrogen bond, you can see the yellow color line, right? So extensive hydrogen bond along the entire neighboring chain. The beta-pleated sheet again is very stable and rigid, means not easily being broken. Oh, sorry, not broken, not easily being bent. Okay. So for example, structure made out of beta-pleated sheet is the silk. Silk is very thin but very strong, okay, because of this beta-pleated sheet structure, extensive hydrogen bond. Okay, so there's a quiz here. Now this is a polypeptide chain, but within the chain, can you identify the alpha helix and beta-pleated sheet? So look for the regular coil and the regular folding. You can find, isn't it? So can you find this regular coil here? So this is an alpha helix, right? There's an alpha helix here as well. Over here, right? So this is the alpha helix here, okay? Another alpha helix here is a short one here, okay? So this is a regular coil, so it's alpha helix. Then beta-pleated sheet is represented by the arrow, flat arrow. Sheet means it's a flat like piece of paper, right? The flat arrow. So there's only one beta-pleated sheet here in this polypeptide chain. Okay, so can you note that the rest of the polypeptide chain, right, it shows no regular arrangement like here. Can you see this is also a polypeptide chain, but there's no regular arrangement like here also. There's no regular arrangement. So the part with the regular coil and regular folding is alpha helix and beta-pleated sheet respectively. All right. So let's try out this question. When hydrolyzed, which molecules have products containing the carboxyl group? So carboxyl group is the COOH group, right? Carboxyl group. Now, phospholipid has got fatty acid, so it must have COOH. Polysaccharides don't have COOH, all right? Proteins is amino acid, so amino acid, the acid part is the COOH, so answer is 1 and 3. Now, question 2. How is the shape of a polypeptide chain maintained when it is coiled into alpha helix? So we just learned that it's maintained by the hydrogen bonds. Okay, you also need to learn out the hydrogen bonds is between which part and which part exactly of this polypeptide chain. So we'll stop here today. In the next lesson, we will continue to explore the structure of tertiary structure of protein.